Hello everyone. Today we will know about the structure of double-stranded DNA. So the Watson and the Crick, they discovered the structure of the double-stranded DNA in 1953. Actually, before that, Franklin and the Wilkin, they discovered the X-ray diffraction of DNA. So from the data of the X-ray diffraction that was discovered by Franklin and the Wilkin, now Watson and the Crick discovered the structure of the DNA in the 1953, and for this they all got the prestigious Nobel Prize except Franklin. Why? Because the Franklin was dead at that time, and Nobel Prize is only given to a alive person. So that's why the Watson, Crick and the Wilkin, they only got the Nobel Prize for the discovery of the DNA. But if in the exam it will be asked that who discovered the double-stranded DNA, then Watson and the Crick. Franklin and the Wilkin, they only discovered the X-ray diffraction data, and from the X-ray diffraction data, now the Watson and the Crick, they discovered the structure of the double-stranded DNA. So now there are different types of DNA. Actually, there is called as the DNA A, DNA B, DNA C, DNA Z. So different properties of the DNA are present, and according to that the naming was present. So most of the DNA are the DNA B. So in our case, or in the human being, or in animal kingdom or the plant kingdom, maximum DNA is the DNA B. So this is the structure of the DNA B structure. So here why it is called the double-stranded DNA? Here two strands are present. Here it is the one strand, here another strand. So one strand is present from the 5 prime to 3 prime, and another strand is present in 3 prime and to 5 prime. That means they are present in the anti-parallel manner. So that means one strand is present from 5 prime to 3 prime, and another is from 3 prime to 5 prime. So in the polynucleotide chain, this is a polynucleotide chain, this is another polynucleotide chain. So different types of the nucleotides are present here. So between the nucleotides there is a bond is formed, that is called the hydrogen bond. That means there are different types of nucleotide, and here the adenine forms 2 hydrogen bond with thymine. So like that, cytosine forms 3 hydrogen bond with guanine. So here the cytosine forms 3 hydrogen bond, and adenine forms 2 hydrogen bond with thymine. So in the previous video we have already studied about the structure of the DNA. So now these nucleotides, or this double-stranded DNA, is present in the right-handed coiled structure. Right-handed coiled or right-handed helix structure. In the previous video we have also studied about what is the left-handed, what is the right-handed helix structure. So this is the right-handed helix structure. That means the DNA is always present in the right-handed helix structure, or it is right-handedly coiled together. So now one is called as the pitch or the turn. So what is the pitch and turn? So this from this to this, from this to this is called a pitch, P-I-T-C-H, pitch, or it is also called as a turn. So in the pitch, the distance of the pitch is 3.6 nanometer. So actually it varies from book to book. That means actually it is 3.2 to 3.6 nanometer. In some book you can be seen that it is the 3.3 or the 3.4 or 3.5. So mostly from 3.2 to 3.6 nanometer. So it is the nanometer. So that means if we, in some book it is also written as 36 angstrom. The same, the 3.6 nanometer in nanometer is equal to 36 angstrom. So the distance of a pitch is 3.6 nanometer or 36 angstrom. So in the pitch there is a major group, there is the minor group. So what is the major group? There is a depression or a huge depression, that is called as the major group, and there is a very slow depression, that is called as the minor group. So now in the pitch 10.2 nucleotides are present in every pitch. On an average 10.2 nucleotides, or it is nearly equal to 10 nucleotides are present. So in exam it will be asked that what is the distance between each nucleotide. So now let's see. So this is the 3.6 nanometer of pH distance, and in the in one pitch 10 nucleotides are present. So that means the difference between the each nucleotide is equal to 3.6 by 10, that is 0.36 nanometer, or it is called as 3.6 angstrom. Okay. So the diameter of the double stranded DNA, that means this one, the diameter of double stranded DNA is 20 angstrom, or it is called as the 2 nanometer. There is a unit, that is the nanometer. Nanometer is 10 to the power minus 9. Angstrom is 10 to the power minus 10. So either 20 angstrom or 2 nanometer is the diameter distance of double stranded DNA. So now sometimes there is called as the triplex DNA. Called the triplex DNA. So what is triplex DNA? And sometimes, that means in the lower pH, that means in the acidic condition, one strand will form and it join to this duplex, and this third strand is called as the TFO, or it is called as the triplex forming oligonucleotide. Triplex forming oligonucleotides. So always this third strand is very small. So that's why it is called the oligonucleotides. Oligo means very small or few. So few nucleotides are present in the TF or third strand, or it is called as the triplex forming oligonucleotide. So now it joins to a duplex, and now it forms a triplex structure, or it is called as the triplex DNA. It is only present in the low pH or acidic condition. So the triplex DNA is sometimes, or it is written as purine.pyrimidine, but this pyrimidine is written as italic. Italic means there is a underline below the pyrimidine. So that's why the PU is written as straight, but pyrimidine is written as italic letter. So this is the triplex structure of DNA. So now come to G quadruplex. So what is G quadruplex? G quadruplex quadruplex. So quadru or quadrant, that means the 4. So what is the G? Why it is called as the G? Simply in the DNA there is a many area in which a guanine is present very rich. That means, let's say this is the structure of the DNA. Let's say this is the A, this is the T, this is the T, this is the A. They are the different types of the nucleotide, but in some region that is present like the C, G, G, C, G, G, A, G. So that is called as the G rich region. That means here a very G or the guanine is present mostly, other guanine rich region is present. So that is called the G rich region. So in that case, so the G quadruplex is present. So why? So in that case they fold each other. That means the 4 guanine structure. So 1 guanine, 2 guanine, 3 guanine and 4 guanine. So they form a G quadruplex structure like this. So here this is the G quadruplex. That means here it is the 1 guanine, here it is the another, this is the second guanine, this is the third guanine, this is the fourth guanine. So they form they fold each other by forming a bond with each other, and always at the center a monovalent element is present. That means mostly it is sodium or potassium. So why the monovalent of the sodium potassium is present? The presence of the sodium and the potassium, they provide the stability of the G quadruplex. So that's why the G quadruplex is present always where the guanine rich region is present in the structure of the DNA, and always in the center a monovalent, especially a sodium or the potassium elements are present to provide the stability to the DNA. So now come to which factor provides the stability to the DNA. So always the non-covalent interaction provides the stability of the DNA. So there are the 2 types of the interaction that provides the stability. The non-covalent can be 2 types. One is called the stacking interaction. And another is called as the hydrogen bonding. So what is the stacking interaction? Look at the structure of the DNA. So this is the 1 polynucleotide chain. This is the another one. So one stands from 5 and ends with 3, and another is starts from 3 and ends with 5. That means they are present in the antiparallel structure. So these are the different types of the nucleotides present. That means adenine, thymine, cytosine, guanine. So they are actually there are 3 types of bond. One is called as this bond. That means in the polynucleotide structure, this bond is called as the phosphodiester bond. In the previous video we have already studied. You can see them. So another structure is called as the hydrogen bond. So hydrogen bond, that means the bond is formed between the nucleotides, between the complementary nucleotides, the hydrogen bond is formed. So another bond is formed between the bases, and this bond is called as the stacking interaction. So what is the stacking, or what is the bond? So the stacking interaction, first one is the van der Waal interaction, another is the hydrophobic interaction, the hydrophobic bond or the hydrophobic interaction. So this is the stacking interaction. That means the bond which is formed between the bases is called as the stacking interaction, and this is the major force or major factor which provides the stability to the DNA. So another bond is called as the hydrogen bond. It also provides the stability, but it is the minor bond or minor factor that provides the stability of the DNA. So the other tip, one is the stacking interaction, another is the hydrogen bond. So van der Waals and the hydrophobic interaction, what is the function of this interaction of the bond? They minimizes the interaction of the water to the non-polar bases. Always the bases, the adenine, guanine, cytosine, thymine, so they all are the non-polar structure. But in the aqueous environment, in our body, mostly water is present. So that's why water is the hydrophilic. So hydrophilic structure cannot interact with the non-polar structure. So that's why the non-polar, the nucleotides, they are present inside it. The outside the phosphodiester bond is formed, and the inside the stacking interaction is formed. So the stacking interaction, the van der Waals force and the hydrophobic interaction, they minimizes the interaction of the water to the non-polar bases. So what is the function of now the phosphodiester bond? So it forms the hydrogen bond with the water molecule. So the phosphodiester bond is hydrophilic, that means they can interact with the water, but the stacking interaction, they cannot interact with the water molecule. So that's why the hydrophobic and the van der Waals force, they minimizes the interaction of the water to the non-polar bases. So this is the two types of the interaction, stacking and the hydrogen bond, and this is the major and this is the minor factor for the stability of the interaction. But once either the stacking interaction of the hydrogen bond is disrupted, now the DNA will be denatured. So what do you mean by denatured DNA or denatured DNA? So that means now the double stranded DNA, this is the, let's say this is the double stranded DNA. So after the denaturation, they forms a single stranded. So this is called as the denatured structure. So how the denature is occur? Either the temperature, either change in pH, or in some chemical, for example urea. So they denature the structure of the double stranded DNA. So when the structure of the DNA is denatured, it can be determined in the UV spectrophotometer. So what is the UV spectrophotometer? In the practical you will be study that the DNA absorbs maximum at 260 nanometer. So let's say this is the graph of the double stranded DNA. So this is the double stranded DNA graph. So at the 260 nanometer, let's say this is the 10, 100, 200. So this is the 260. At the 260 nanometer, what is the nanometer? That is the wavelength. So at the 260 nanometer wavelength, the DNA absorbs maximum. So that's why this is the absorption maxima of double stranded DNA. So what is the how we will know that the DNA is denatured? If the DNA is denatured, that means if the stacking interaction of the hydrogen bond are disrupted, then the absorption maxima increases very much. So this effect is called as hyperchromic effect. Hyperchromic effect. So that means when the DNA is denatured, the absorption maxima increases very much from that of the normal one, and the effect is called as the hyperchromic effect. So one is the hyperchromic effect is found if the DNA is denatured, and again the viscosity and buoyancy decreases. So that means once the DNA is denatured, one effect is the hyperchromic effect, that means the absorption maxima increases, and another is the viscosity and the buoyancy of DNA decreases. This is the effect of the denatured DNA. So this is the all about the double stranded DNA structure. So if you like, then please like and subscribe my channel. It will take maximum 2 seconds or the 1 second. So please subscribe. Thank you.